about 7 years ago. Right. And before that, he was assistant professor at the National Tsinghua University in the Institute of Biomedical Engineering. And he told me he really glad to move to INS. He really liked it here because we have better facilities here. So this allows him to do cutting edge research. Okay.
Better people.
Okay, and the better people, of course. So that's what he is going to tell us, how he can do those kind of research in this institute. Let's welcome him.
Thank you for coming on a Friday afternoon. I think if I could turn off the light. I have the home theater advantage. I know this place quite well now. So again, thank you for coming on a Friday afternoon. I know that you probably have other things to do, and the fact that Dr. Mo is also here, thank you for your presence. We're very honored to have you here. So, though this is my quote-unquote promotion talk, I still treat it with utmost respect, and it really took me a lot of thinking to come to this title, which is from cells to systems. And in terms of that, I approach it in a multi-resolution manner of molecular bioimaging on extracellular vesicles and DNA repair. So before I go on a little bit further, I will just go over how I usually approach my research. This is my lab's name is Bio-Nanotechnology and Molecular Bioimaging. Essentially, we combine imaging, biology, and application together. And hence, most of the work that you see from our lab is at this sweet spot. If it's only these two or these two, it usually doesn't make the cut. And the slogan that we have is that we create and apply new technologies to explore biological phenomena. And since I've been tasked to give the INS BPTQ vision statement, this is essentially what we come up with, is to develop key methods to explore important biological questions. And to break it down further into our mission is to first develop key methods and to identify the important biological cues. And this is how, at least I practice what we pray. This is what I try to do ever since I started my lab. So for today's talk, I will focus on two, at least what I consider as important biological questions. Part one would be on extracellular vesicles. And I think in Taiwan, this is what you know me by mostly. On the other hand, for the latter part of today's talk is on DNA repair. So you probably don't hear as much of me in Taiwan when I present this, but I'll go over it really quickly towards the end. So here's part one, is to unveil vesicle network and dynamics in vivo. So again, I know that people come from a variety of backgrounds, so if you don't mind me, I'll try to make an extended metaphor. Cells are well connected, and so are our world today, that we are connected by different networks, be it direct networks, such as if you have different computers and modem and servers, they are connected by Ethernet, or the undersea cable to connect major networks around the world. At the same time, there are many wireless networks that connects devices such as what you have in your hand, the 4G, 5G, or Wi-Fi, or whatnot. Can you imagine if you don't have these networks? Think it for a sec. I think the world would be very different. You can fly drones. You can do blah, blah, blah. So how about cells? So in fact, cells are always connected one way or the other. And this is biology 101, because I know people come from different backgrounds. This is a central dogma of cell structure, that you go from subcellular or cell to cells, then to tissue, then to organ, and if you have organs working together, you call it organ systems. Then finally, you will form an organism. This is how you usually break down in biology 101. And since my basic training when I was, not when I was a kid, when I was young, my background is a biologist. So when I look at a problem, this is what I think about. You cannot convince me to only look at subcellular or subatomic without thinking about what really happens here and here. And hence, I'm hoping to convince you that this is how you usually approach the question, is that how do they communicate at different scales of these systems? Moving forward, how about the networks in our body? So for cell biologists, we do know that cells actually do form physical conduits. For example, gap junctions, such that they form direct channels between the cells to allow them to pass different molecules, such as ions, as well as nucleic acids. And gap junction was something that I used to work on when I was younger as a PhD student. And later, people start to find, of course, there are proteins and receptors that the cells express to facilitate cell-to-cell communication. Until about the early 2000s, people started to find that there may be other ways of how cells can contact with one another without touching each other. Okay, so they don't need to shake hands, but could they just like have wireless network where they can send signals or packets of information out to allow the cells to communicate? And come along extracellular vesicles. So the term that you probably hear mostly in Taiwan's market or the academic field would be exosomes, but to be academically correct, exosome is actually just a subpopulation of what we call extracellular vesicles. So essentially these are vesicles released by cells to mediate cell-to-cell communication. If you categorize just by the size, they range from 40 to 100 nanometer, we call them exosome. You can categorize them into exosome small or exosome large. And then if you have bigger ones, you can have microvesicles that can be up to 1 micron in diameter. Now this is TEM image to show you the canonical lipid bilayer structure of these vesicles isolated from the fluid released by the cells. And of course, there are some bigger ones that can be up to 4 micron that the cells release as they die due to apoptosis. And they're quite interesting because they do carry DNA fragments, whereas most others don't typically do. Now there are also very big ones released by cells. They coined the term oncosome for the Bezos group that can be up to 10 micron. So if you imagine that a cell typically is about 30 micron in diameter, 10 micron is pretty big. Now, of course, other scientists from China, Tsinghua University, not the one in Taiwan, the one in China, they also found that cells can release like a pomegranate-like structure for the subsequent release of the vesicles within, and they coined the term migrosomes, which help cells migrate as they develop. And there are also what we coined the term bio-nanoparticles. So they don't have the lipid bilayer. They are essentially just particles, and they're very tiny. They're less than 50 nanometer in diameter, released by cells to also facilitate communication. So this is cartoons to show you how those vesicles or particles facilitate intercell communication. In this cartoon, the donor cells can release vesicles through the fusion of multivesicular body to the plasma membrane to release what we call exosomes, or through the shedding of the plasma membrane, such as ectosome or microvesicles, or through yet identified mechanism to release nanoparticles. And all of these carry bioactive cargo, including proteins, nucleic acids, and lipids that can be transferred to recipient cells and offload these cargo to communicate with the recipient cells without touching one another. And this is actually how Wi-Fi and our network work. They don't have an invisible line that connect to your device. They send information in packets. And lo and behold, cells have been doing that for, I don't know, centuries, millions of years. And so can you imagine when we first found that this actually exists to biologists? It's like it just completely expanded our horizon of understanding as to how cells communicate. And moving forward, and of course, as you will know, disease usually hijack normal processes in the cells to their advantage. And of course, that's the case for cancer. Cancer have been reported repeatedly to release vesicles before metastasis to educate pre-metastatic niche. So if you don't know about cancer, I trust that a lot of you watch soap opera, and then the 主人就是主角 they're gonna say that, oh, I'm dying due to cancer, so why? And you're gonna go, well, I have terminal cancer. And terminal cancer usually means the cancer cells have metastasized from the primary site. And usually it's less easy to control, but with the advent of biology, it's becoming more and more controllable. But cancer have been found to release those vesicles to facilitate the metastasis process. And of course, in terms of neurological disease such as Alzheimer's, Parkinson's disease, vesicles have been found to play a role to promote the disease progression. And this is important as our society is aging, and disease such as Alzheimer's, Parkinson's have been found to associate with vesicle release to the surrounding region to form, say, the amyloid plaque and such. Heart disease, people have found that vesicle play a role in heart cardio protection or cardio damaging. And don't underestimate the effect of heart disease. It is actually the #1 cause of death in the United States, not cancer. Yeah, so a lot of people look into that. And then immunity, people have associated vesicle with infection. Those things, due to the time of today's talk, I'm going to go into detail, but a lot of people are looking into using vesicle as a biomarker because they carry these bioactive cargo inside this nicely shielded lipid bilayer. And essentially what they're hoping to do is to do what we call liquid biopsy. What it means is that you can do diagnosis based on, say, urine, blood, and you can do diagnosis or even do monitoring. So this is a lot better than, say, going to the doctor and then have all the examination. All you need is just pee or provide the feces sample, and then you can do diagnosis. That's what a lot of people have been working on. On the other hand, scientists such as us are trying to use that as a therapeutic vehicle. they're hoping that they are more endogenous, meaning that they're released by cells and not as foreign to the immune cells. And there are many senpai or former giants that developed things such as synthetic lipid particles that are trying to carry therapeutic cargo for therapy. To my knowledge, unfortunately, a lot of them still could not work due to the very different immune system between mice and human. And hence, a lot of people lay their hope onto vesicles because they are more endogenous. And they're hoping that they could be a more effective therapeutic vehicle. Now, if they are basic scientists here, biologists, they are also a small group of people trying to understand how vesicles play a role during development. But for the purpose of today's talk, I'm not going to go into that. All right, now how do you detect vesicles, right? So imagine there are so many wireless waves between us. That doesn't mean that they don't exist just because you don't see it. Similarly, for the vesicles, they actually do exist everywhere in the body. How do you detect them? And that has been the biggest problem. Most of the method in detecting them is by using, say, for example, microscopy, including SEM, DEM, AFM, or you can measure the biophysical properties such as dynamic light scattering, nanoparticle track analysis that relies on, just to name a few. Molecular biologists such as we will use molecular cloning techniques to tag fluorescent protein to some vesicle markers such as CD63. On the other hand, chemists, they will use, say, antibody to label some vesicle markers such as these tetraspendin listed here. Or you can develop dyes, especially lipophilic dyes, to label this membranous vesicles. And these include a commonly used PKH dye and so forth. And this is just SEM of a really highly aggressive brain cancer to show you that when cancers arise, they have all these vesicles on their surface ready to be released. And this is a classical image, image by one of my former colleagues. So when I started working on vesicles, to be honest, was not a believer. It's like when you first see cell phone, do you actually believe that I work? It's good for self-defense because it was very heavy. 以前有一个黑金刚, right? 一个可以用来当砖头, to protect yourself. But to me at that time, I could not believe that this actually exists. So I thought, let me try to visualize it. So one of the most common dye used to date is PKH dyes, and then essentially they're very long aliphatic compound that has long hydrocarbon chain that has a half-life ranging for 100 to a week. And at the time, I kind of go, well, this is weird. I don't think vesicle can last that long in the body. If that's the case, then everybody will look like the sumo. Okay, it just doesn't make any sense. And not only that, these aliphatic compound, essentially they're lipophilic, they actually automatically form micelle. Essentially, they form nanoparticle by themselves, even without labeling the vesicles. And not only that, just like coloring your Easter egg, you need to get rid of the dyes after you color them, which is also problematic because the yield then of the vesicle that you label is extremely low. You lose at least 30% every time you wash it. And similar principle apply to other lipophilic dyes. And moreover, the labeling efficiency, such as how well the dyes color the egg, is also a problem. So I set a couple of criteria when I started my studies. Could I potentially generate something to hand label the vesicles? Because as you would remember, the vesicle come in different sizes and through different biogenesis pathways. And in terms of accuracy, could I achieve spatial as well as temporal accuracy resolution? And finally, in terms of resolution, I was hoping that I think I was young and reckless. I just thought, hey, I want to see the vesicles move around in the whole animal. And further, can I actually then, on the other hand, look into subcellular resolution? So remember the cell to systems idea that I was mentioning about. That's what I said to do when I was doing my postdoc back in Mass General Hospital and Harvard Medical School. And we were very fortunate to generate one of the first bioluminescent-based reporter to track vesicles in vivo and further apply this to study melanoma release vesicle and how they interact with the immune system before they eventually overcome the immune system of the body and educate the immune system to their own advantage. That was back in 2015 or 2016. At that time, I also developed another series of fluorescent-based reporter that labels the inner leaflet of the lipid bilayer by using palmitoylation moiety molecularly conjugated to fluorescent proteins such as GFP tomato. And under live cell confocal microscopy, you can see the vesicles release or budding off from the cell surface. Yet at the same time, you can also see the vesicles release to the surrounding region of the cells. So now I look back, this is like kind of my former life. So when I started my position in Taiwan, I thought, wouldn't it be nice if there's one reporter that can image vesicles from vesicle resolution, essentially 10 to the negative 9, to cell or tissue resolution, and then to organ, and then finally to whole animal. And I think the idea that came to mind is Google Map. I just use it literally every day, and actually the fact that I actually made it back after traveling to Thailand and Tokyo was because of Google Map. And light came to mind. The method that, but this is just sort of showing you the real world application. I was visiting the University of Tokyo ArtCast, and then I was trying to find where they are, and I was in Shinjuku. I literally had no idea how to get there. I went to the Metropolitan Tower, the 2 tallest tower in Shinjuku, and I looked at it. I was like, without Google Map, I would just be lost. Because think about the Densha station they have, it's so complicated. And wouldn't it be nice if we have a Google Map like tool for vesicles to tell us where the things are moving? Because this is critical to understand how they work in terms of biology and how we can reverse engineer them for therapeutic applications. And the idea came to mind is to use something called bioluminescence resonance energy transfer. Most of you, I think, 老师知道 Fred a lot better than I. Yes, but what I meant is that yeah. But instead of FRET or a pair of fluorescent reporter, it's a pair of bioluminescent reporter molecularly conjugated closely with a very short linker EGFP. In this setup, all you need is to treat them with substrate fluorimazine or fluorofluorimazine, the most updated version. In the presence of this cofactor oxygen, through the oxidation process, it will emit the peak signal at 450 nanometer. At the same time, this signal will also, through resonance, bioluminescence resonance energy transfer, to excite EGFP to emit a signal at 510. If you're not satisfied with the intensity, you can also externally excite it to also give you this 510 fluorescent signal. So this setup actually has 2 signal output. One is the bioluminescent 450 nanometer, the other one is the 510 fluorescent signal. And similarly, we use a palmocylation moiety to label the inner leaflet of the lipid bilayer. In such a fashion, the cells, the membrane inner leaflet will be labeled, and so will the vesicles released by them. And through this method, we actually achieve what we set out to do. We were able to track vesicles at whole animal resolution through organs, tissues, cells, and further, by using super resolution right out fluctuation calculation, we were able to achieve super resolution imaging of the vesicles. And here you can see that we overcame the diffraction limit and can actually image individual vesicles. So those, I'm sorry, I don't have a magnifier, but you can see 2 dots rather than a blur. And this was very important because essentially what we achieved is multi-resolution imaging of the vesicles with bioluminescent or fluorescent quantification, which is critical when you're trying to study stuff in vivo. If you can't quantify, you are just doing qualitative observation. And quantifying nanoparticle in vivo has always been key in this field. And with this, we were also able to do very sensitive biodistribution analysis. Biodistribution is essentially studying where the stuff is going in the body after you administer them. And this is a really critical part of biology as well as pharmacokinetics. Pharmacokinetics is what how your body treats the compound. Pharmacodynamic is what the drug does to your body. So this is a really important part of pharmacokinetics. And further, we identify that vesicles actually has a very dynamic distribution in the body, such that if we change the organotropism, meaning that if we change the direction of the vesicles going to certain organs, they actually go somewhere else. Previously, people could not detect this due to the poor detection limit that they have. They could only look at major organs such as the lung or the liver. So in this case, we actually show that if they don't go to a certain targeted organ, they actually go somewhere else. And this is really critical in our understanding of how these nanovesicles work. And we also did Journal Club. This is hosted by International Society for Extracellular Vesicles. This is the biggest and the largest vesicle society that we typically go to annually. And during COVID time, they started hosting the Journal Club. And if you're interested, please feel free to visit. This is what I looked like when I was young. And this is what my PhD student used to look like when he was young. He's now a postdoc finishing up his work here. Moving forward, we were able to be covered by Nature Methods and further apply this through collaboration with Dr. Li Guoping. And at the same time, our method was applied to study one of the first pharmacokinetic study in non-human primate. And we were very proud of this because most of the study model is like, for example, mouse or swine. 就是猪啊，不是那个天人，yeah. And then in this case, we actually were able to collaborate with Dr. Kent Whitworth from Johns Hopkins Medical University to apply our reporter system to study how the vesicles move about in non-human primate, and then reported this in 2022. So moving forward, as I mentioned before, vesicles do come in different types or biogenesis pathways, and they come in different sizes, and there were just so many of them, it's really hard to narrow down. We kind of just say, you know what, let's forget about how they were generated, but we definitely know that they are different sizes, and we categorize in such a manner it's easy. Anything less than 200 nanometer, we call them small vesicles. Anything greater than 200 nanometer, we just call them big vesicles. Okay, that's simple enough. We wanted to use the word large, but at that time the terminology is still quite a mess, so we just call it big. And then what we found was this, that we started looking to breast cancer cells. So this is just a really quick overview of breast cancer. They come in different molecular subtypes. There are the luminal ones, such as the HER2 positive, ER/PR positive. There is the HER2 enriched, and there is the triple negative. Essentially has no major marker. They're HR, ER/PR, and HER2 negative. And triple negative breast cancer is one of the most highly aggressive and hardest to diagnose cancer among the many molecular subtypes of breast cancer. So we begin to ask the question is that, do the different molecular subtype of breast cancer have favorite release of specific vesicle population, say for example the big or the small? And do the different vesicle populations, say the small or the big, do they confer tumorigenic activity? Basically, do they support tumor growth? We didn't know about that. We do know the small ones will do, but we don't know about the other ones. So we went to test and actually found something interesting. By using nanoparticle tracking analysis and by examining a panel of breast cancer, we actually found that, so M10 is the control, this is a normal epithelial cells. We found that, and then the blue line is the big EV, the orange line is the small vesicles. By looking at all the triple negative cell lines, we actually found they release more big vesicles when compared to the control here. And then the ER/PR positive molecular subtype also release more big EVs. And then the HER2 positive and the normal epithelial cells actually release predominantly SCV, the small EV, but not so much the big EV. And by doing the ratio between BEV and SCV, we actually saw this trend quite clearly, that all the triple negative breast cancer cells release a lot more BEV than the M10 epithelial counterpart. And then moving forward, we actually did some questioning. It's like, wow, could it be that cells do release, especially cancer cells, release different vesicle types? And this is just to let you know that about in the past almost 20 or 15 years, majority of the study only looked at the small vesicles. They only looked at anything around 200 nanometer or less. Frankly, I don't know why, but I think I know why. It was because all of their former senpai always tell them to filter their media with a 0.22 micron filter before isolating the vesicles. And one day, my student Brian and I were chatting, I was just like, dude, I really don't think cancer would rely on one vesicle type to do their deeds. Why don't we just remove the filter and see what comes out of it? And this was one of the first findings that completely surprised us. And moving forward, we started to examine by using a 41, this is a mouse triple negative breast cancer cells. We also isolated their big vesicle and small vesicle, ran it on their TEM, and we did image J and did calculation from 30 images with 400 particle count. We can see a very distinct distribution difference between the big EV, which has an average around 300 nanometer in diameter. This is one time, okay, this one's okay. Yeah, and then we found that the SEV actually is smaller, around 100 nanometer in diameter in the means. Yet the distribution is just drastically different. And by doing compositional analysis, we found that big vesicle primarily consists of 200 to 400 nanometer vesicles in size, whereas the small vesicles consist primarily from 100 to about 200 nanometer vesicles. And this was essentially what sealed the deal with the reviewer, or else they could not believe what we were claiming. And we thought, okay, that's really interesting. The cells, cancer cells actually release different size vesicles. How about let's try to track them? And at that time we were, I think being Asian and being Taiwanese, we're very humble. We still believe that the dyes are better. So we thought, why don't we do this? We express these vesicles with our reporters, the Palm Grad reporter that Anthony developed. And then let's label it at the same time with a dye. And that's what we did. We used most of the common dyes people use for in vivo imaging, including PKH, DID, DIR. This is more into the infrared spectra. And then essentially what would happen is you have the big vesicle with double signal, and then the small vesicle also with a small signal. And we started doing in vivo study to say, let's inject them at different doses, either BEV or SEV from 10 to all the way to 100 microgram through tail vein injection. And then after injection at half an hour or 24 and half hour later, we sacrifice and collect their organs. And we want to see how the signal go. We were putting a very high hope to the dye signal. Unfortunately, something happened. Anything injected with 100 microgram animals, they die within a day. Anything injected with 50 microgram, most of them die within a day. And then the only one that survived is around 25 microgram, and the 10 microgram survived completely in the big EV category. And the one that only survived really well is around 10 microgram. And this took us by complete surprise. Before this, no one ever reported that the vesicles can kill. Okay. And funny thing is that we were so scared in publishing this, but we just kind of say, this is a fact that we need to tell people. And after we published it, people come to me and say, yeah, we found the same thing as well. And I'm like, why did you publish it? I was like, well, it doesn't add up to the paper. Well, come on. Like, so anyway, now this is a commonly recognized fact, but I emphasize that this happened mostly in 4P1 derived cancer cells. We found that other cell types don't release such as sticky vesicles. They may be sugar. We don't know yet, but we just want to point out that dosage is really important. Reason being is this, that I'm gonna skip over this part, is that a lot of people are trying to find really good signal. So they drive up the dosage until we found that the animals died. Yeah, and in this case, we're trying to tell people that, hey, I know that although it may be safe with poor signal, try to find a common ground, like between the safe and good signal for your study, because it's what we would call a more physiologically relevant study. Okay, so I kind of skipped the last part, but due to time, I'm gonna kind of move forward with it faster. So after we got the dosage right, so which is 10 microgram, we went to identify what I mentioned before is the organotropism of the vesicle. We actually identified some cancer-associated membrane protein that directs where the vesicles go in the body. We found them by using mass spec and proteomic analysis, and there were a couple that we picked, which is SLC29A1, CD9, CD44. And then by using molecular methods, we knocked down the expression, and then we isolated those vesicles to put into animal and see what happens. And what we found was actually quite interesting. Be it the big or small vesicles, when we knock down the expression, and in this case CD44, they actually don't go to the lung as much. Okay, so I'm sorry, this is a little bit busy, but I'll try to guide you through quickly. This is our control scramble with big EV. This is a pie chart distribution of the vesicles in the major organs. A lot of them went to remain in the blood, but after 18 days of education to the mice, a lot of them went to the lung. If we knock down the CD44 membrane expression, the amount that went to the lung drastically reduced. This is the case for big EV, and similarly for SEV. Again, 31% dropped down to 13%. And usually this is where people end their story. But no, with our reported study, we actually show that when they don't go to the lung, they actually go somewhere else. In this case, they remain in the bloodstream. Okay, and then we also further found that small vesicles seem to go to the spleen a lot more than the big vesicle. So the way of how the big and the small vesicles distribute is actually not the same. And this was the first study to systematically compare the big and small vesicle in vivo property in the world. And moving forward, if you're interested, sorry, I'm doing like YouTuber, but if you're interested, I'm not a YouTuber, but to our surprise, this is one of the highest viewed video on this channel, which is only a pathetic 8,000 plus. But the fact that there are 8,000 scientists that view it, well, okay, maybe they view it twice, 4,000 times that view it, we were very happy. Yeah, so Brian did an excellent talk here, but as a really quick summary, we found that the dyes can affect the label vesicles in terms of the size, and the spatial and temporal resolution were completely off. we also found that EV dosage is critical, so we raised this concern to the general public. We found that both vesicles, the big or small one, are actually tumorigenic. They do both promote tumor progression or tumor growth, and they actually exhibit distinct biodistribution pattern at organ systems level. It's not just the lung or the liver, it's actually the whole entire system change that we should be looking at. And further, we identify these organotropic proteins, which may be reversely engineered to target certain organs. So the significance of this study is such that people have been looking at small vesicles only for the past decade or so. So could it be that the big vesicles also play a role in diseases? Again, I kid you not, they literally just because of that filter that has hindered their study for the longest time. If I may speak in Taiwanese, 那个人家说啊，你拿那个把酒叫喇叭勾掉，啊，你是讲你撒喇叭来勾把酒。So I'm just saying that sometimes don't just follow your former colleague, just think a little bit further ahead, and maybe you will see something new. At least we were very lucky. And further, could it be that both small and big vesicle could carry different bioactive cargo for subsequent diagnosis? And at the same time, could it be that the vesicle, the bigger ones, can be such as a heavy bomber, because we know simple math, they have a greater surface area, their volume is greater, that they can carry more cargo, so they can deliver a lot more. But yet at the same time, they probably don't carry as far. So they still need the small ones to carry them to the tumor, such as through the EPR effect. So those things needs to be examined further and compared. And with this, we're very fortunate to start the first large vesicle workshop under International Society for Extracellular Vesicle this year. And then I was one of the IOC, and this took place in Columbus, Ohio this year. And I'm hoping that people will start looking into both of the different sizes of vesicles from here on. And moving forward, then, you now know that there are vesicles, there are the bigger ones and there are the small ones. But it always pains us that we can only look at one vesicle population at a time. It's like you have all the different wavelengths, but you can only detect one at the same time. So it's similar to Pei-Ling's study yesterday. Is I love the metro idea, and it has stuck with the most. It's like we can only look at one. So could it be that wouldn't it be nice if we can look at different vesicle populations simultaneously? Because we now know that BEV exists, we now know that small EV exists, and but cells do release all of them at the same time, right? It's not like they only release this at the same time at once. So we just want to do simultaneous tracking, and we believe that this would give a more physiological relevance in our study. And hence we developed a second pair of REC-based reporter. We coined the term POM-SORET. And the setup is very similar. It essentially has one bioluminescent reporter called Raniella luciferase, and is closely conjugated, molecularly conjugated to IRFP. In this manner, when you expose this bioluminescent luciferase with bottle blue, you know that this guy drank a lot of coffee because they want to coin blue bottle because of that heat, right? Yeah, So, but with the presence of oxygen, it will emit the peak signal at 415. Through SORET excitation, it can actually excite RFP, so to emit a signal at 750 nanometers. If you're not satisfied, similarly, you can use external excitation wavelengths to also drive the fluorescence signal. And again, we use pulmonary signal to label the inner leaflet, so that the inner leaflet of the cells is completely labeled. I dare not say complete, mostly labeled with our reporter. And this pair took us about a year and a half. We have tried many pairs, at least 8 combinations, to find one that does not have very significant signal crosstalk. And essentially, this is a cartoon to show you what we initially planned to happen, and I think we made it happen. So that if you treat it with bottle blue 2.3, you will have the we label the big vesicles with this POM-SORET, and then we label the small one with the POM-GRET. And in such a fashion, if you treat them with a substrate, when you treat it with this substrate, the small ones will light up. And when we multiplex it, we coined the term POM-SORBET. Sorbet, this is a small one. The Italian ice cream, yeah, that doesn't have cream. Yeah, so if you're Italian, you appreciate this a little bit more. At least when I gave a talk, they were laughing. But moving forward, we decided to do proof of principle to show that when you have co-culture of both cells, you can see this is again POM-GRET, and the purple one is POM-SORBET. But you can see the exchange of the vesicles in both cells, the green in the purple purple, and sorry, it probably doesn't show that well, but the green ones actually have purple inside as well. So this tells us that, okay, in terms of fluorescent imaging, it's spectrally separated, which is fine. And we thought, why don't we try to see if they have significant signal crosstalk? So we first established a standard curve by by seeding different amount of vesicles, ranging from 0 point something to about 1 point something microgram. And this is RIU, this is a bioluminescent signal detected. And we saw a very nice standard curve with an R-squared value of 0.9983. We mixed the big and small label vesicles with different ratio between 0.3 and 0.7 or reverse. And we found then that even when mixed at different ratio, they align very well with the standard curve. So this tells us that this reporter, the palm Sorette, does not crosstalk with the palm Grep. And then we did the reverse experiment where we treated them with fluorophorimazine to show up the small vesicles. Again, we established the standard curve with 0.988 R-squared value. We mixed the vesicles similarly to the one you see on the left with different ratios. But again, we saw the signals detected in the mixture very well, aligning with the standard curve. So this gave us the confidence to actually do animal experiments. And we found that when we co-administer the big and small vesicle at 50 microgram, again, I mentioned this is 293T, they can tolerate 50 microgram okay through the tail vein. And then 4 minutes following tail vein injection, we were able to image it by using IBIS, where the big vesicles are going. And then we thereafter treated them with fluorophorimazine to light up the small vesicles, and we also saw where the small vesicles are going. And after that, about 30 minutes of the injection, we took the organs out, and then we treated them with a blue bottle, and then we found the vesicle distribution ex vivo. So these are the distribution of the big vesicles. Again, these are same animals. Yeah, and then we found that when we treat them with fluorophorimazine, we actually found where the small vesicles are going about. And this is extremely important to us that we can now track both the big and small vesicles at the same time. And what we did next is that we lowered the dosage down to try the detection limit, because we were worried that we always had to administer a large dose. So we lowered it down, we injected, and then sacrificed the animal 30 minutes post-injection to do what we call biodiffusion analysis. Essentially what we're trying to see is that if you co-inject it, do they exhibit differential distribution? And here we actually show that the big vesicle predominantly were detected in the lung, whereas the small vesicles mostly are still in the lung, but the distribution actually is a little bit different. The small ones appear to be in the liver a lot more when compared to the big vesicles. And this is proof of principle experiment to tell us that we can now move forward to apply this multiplex palm sorbet system to study how the big and small vesicles work under pathological or physiological environment. So as a quick summary, we have developed this palm sorbet multiplex vesicle reporter system that can label the different vesicle populations, and then we can perform biodistribution analysis both in vivo or ex vivo. And further, we can actually do super resolution imaging. Essentially what this allows to do is orthogonal analysis to cross-validate the findings and see if there's a consistent trend in the findings, which is extremely critical in the vesicle field right now. People nowadays in the vesicle field don't just rely on one way to tell them that this is right. We actually do 2 or 3 sets of different methods to prove the same point. And then with future direction, as I mentioned, we're hoping to apply this multiplex system to understand if vesicles in different populations have synergistic or antagonistic effect, for cell biology of the vesicles, or for therapeutic development, right? There are drones, there are missiles. Are you just going to send one of them, or are you going to send them both, right? So those are things that we're looking into right now. All right, so for future directions, because this is a public talk, I don't try to reveal too much, but we're trying to develop what we call EV spatial biology. So something like this is the front door of Academia Sinica in Nangang. What if this is a tissue, and if this is a cell, I'm trying to find these cells, and I want to explore what these cells happen after they receive the vesicles. Okay, and then essentially we're trying to develop what I call the vesicle Google Street View. We're going into below this resolution to see what happened after they start receiving the vesicles. And because I think this is important to identify, we now know where they go in terms of organ, we also know which tissue they go to, but we don't know what cells exactly took them up, and what happened after these cells took them up. And that's what we're exploring right now. And this is, I'm sorry, a lot like election. Okay, they receive information, eventually the outcome come out. And I was in Thailand when I heard about the US election. I'm like, wow, the world is very interesting. Yeah. So moving forward, I'm also very interested. We're working on cross-kingdom vesicle communication, such that we're looking into how mammalian cells interact with other kingdom organisms, such as bacteria or plants. That's something that we're working on, but for today's time, I won't have time. But I'm going to spend the rest of my time on the second part, which is much shorter. It's on vesicle. Sorry, it's on DNA repairs. Okay, sorry. This is really quick. A lot of you probably is familiar with cancer treatment, one way or the other, from soap opera or personal life experience. But gene therapy or cancer treatment alike, they essentially, or most of the original treatment relies on DNA double-strand break. What happens is that, as you know, DNA constitutes double-strand, and you can actually cut them by using, say, gene therapy tools. This is a very old technique called meganuclease, thin finger nuclease, TALEN, and of course the most, the latest CRISPR-Cas9. They started doing double-stranded cut to basically break the double strand. On the other hand, radiotherapy or chemotherapy, they actually do similar to break down housekeeping genes. Okay. Now, cells are not dumb. They have evolved through evolution to repair them, and there are two major repair pathways. One is what we call homologous directed repair. The other one is non-homologous end joining. To make it simple, HDR is like copy and paste for your Word file. If you have a template beside it, it can just copy and paste the entire file to replace the broken part. NHEJ is like writing gibberish. You see there's something wrong, the cells will repair with gibberish. They will add or delete nucleotides. This is much preferred by science engineers because they want to insert functional, healthy gene to cure diseases. And they don't want this because if the DNA is randomly repaired, it might cause cancer or some other crap. So earlier on, when I was starting my position, I developed one of the first detection systems relying on bioluminescence. In short, we can actually, without sacrificing the cells, by expressing what we call blur or bioluminescence repair reporter system, when they undergo NHEJ or HDR, they will release respective bioluminescent protein to the media or the biofluid, such as the blood, and you can determine if they are doing one more repair than the other. And this dynamic is actually very important in determining the efficacy as well as the outcome of therapy. So I'm going to skip this really quickly due to time. But I think just by chance, like we didn't mean to call it blur, but it just became that. But we thought, why don't we try to create one that's better? Okay, so as you could tell, bioluminescence doesn't have a very good spatial resolution. Okay, the signal is like, again, it's a glow. So we thought, wouldn't it be nice if we can have spatial resolution on top of what we have already achieved with the blur reporter? And this is what I mentioned before. You can do bulk detection, but you cannot do single cell detection, and nor do you know where it actually happened in the tissue or the cells. So we want to combine all the pros from the blur reporter. We also want it to be non-invasive, and we also want to be able to tell which cell is actually going through the repair with that or that. Okay, and with that, we also confer spatial as well as temporal resolution. Again, we don't need to sacrifice the animal nor the cells. Just by detecting the reporter signal, we can tell what's going on. And we can further isolate them for subsequent analysis. And we have been doing this. We went into some kerfuffle throughout the way, but we're making steady progress. So I'm talking about this to my grantee. We are still working on this. We're making progress. But essentially, we have now confirmed in terms of in vitro assay that yes, we can do this in vitro. We can actually do fluorescence microscopy. We can even sort them out, and we can actually do DNA sequencing to confirm that our sorting is correct by using this better reporter. We're testing this on breast cancer cells to study how the different mutants of breast cancer respond to anti-cancer compound by using different DNA repair mechanism. But this is too bio, so I'm gonna skip that. And I think I'm doing A-okay on the time. Finally, I think I have some home game advantage since I rarely give a talk in our home turf. I'm recycling my slide. This is from 3 years ago. I asked this is my daughter, by the way, in case you're wondering. Yeah, and I asked her what is an odd number, and she says a strange number. I'm like, you're not wrong, but that's not how you define it. And I told her that it's an integer number that cannot be categorized into groups of 2 each. And I think that defines me quite well, because when I first came to IMS, I think I'm an odd one. You know, but throughout the years, I'm learning, and I mean, to be honest, when I first hear about Hamiltonian and ab initio, I'm like, what? And Alexander Hamilton was actually the first thing that came to my mind. And over the years, I think I also try to convince you that odd is an oddity, and it's kind of unique, independent, maybe specialized. I guess quirky is a better way to describe myself, and by quirky, I mean maybe I have some unusual, interesting or appealing attributes that allow me to work with you. And I'm hoping that throughout my years here, I have been collaborative, and I hope I contributed a little. And I hope, actually, this is something I want to say. 我上次这个梗没有抓到. I'm hoping that I'm at least a quark of IMS. And then I saw my kids, and they were just like, yeah, you know how neutrons are. I don't know what is up, what is down, and I don't know what is charm, but that's as far as I go for the quark. Finally, I know I shouldn't say this. I just told the director I feel I'm aging. He said that I'm older than you. And I was like, okay, I think I'm growing. But along the way, I always feel that, as the title says, that I still haven't found what I'm looking for. This is actually a very nice song back in the days for all you youngsters back there. Still a very classic song, so check it out. It's very nice by Bono, YouTube. He's still active. But I guess what I meant to say is that throughout the way, like, I'm still trying to find better ways to approach the research. I mean, to put it more simply, is are there better reporters? Are there better instruments? Better non-labeling? And I think those are really cool. Jalo and I talk a little bit about those. There's many things still being ventured on. But I think as I mature, I'm still trying to find better biological questions. And this is still an ongoing trend that I do every day. I mean, it's kind of painful, but I do every day. And I think that's what makes it fun. But I just really like this. Daft Punk it used to be a really popular band that they disbanded automatically because according to them, they found that AI can one day do better than them. They actually quit way earlier. And I really like it because their song is like harder, better, faster, stronger. Science is always like that. But my question is, but where? And I love the Japanese translation is that Shikoto wa owaranai means that the job never ends. I guess that aligns with scientists quite well. And then I was just in Thailand. I posted this picture, some of you might saw. And my colleague, and then the sentence I put on there is that I was spending great time with awesome friends. And my friends was like, where are your friends? It's just missed. And what I meant to say is that along the way, I found that research is much like this. You probably don't see things very clearly up ahead, or you just can't see it, but you try your best. And you do have friends. They are there. They probably don't show up all the time. And sometimes, I was very lucky, to be honest. I usually bring an umbrella with me. And this, you might recognize this Li Qianming from Academia Sinica. He didn't, I'm sorry, you can tell him, he was not very well prepared. It was freaking cold that day. It was like 10 degrees. And I was like, I have an umbrella, you want it? He was like, great. And then see this towel right here? This towel was from my hotel that I brought for swiping my sweat. I was like, you probably want to take this towel, but just remember to return to me. So I was very happy, and he was such a great guy to travel with. And so along the way, he helped me, I helped him with the local tour professors. So it has been a very pleasant trip, although it seems very foggy and gloomy. But after we made it to one of the tops, as we came down, we actually saw a line of trucks or like a bus of people trying to get onto the mountain. So we actually went up the mountain around 4 a.m. that morning. And then, so this is much like research. You're usually the pioneer, the excavator, trying to challenge its frontiers. And although the outcome or the forecast is not that positive, but we do our best. And we take a break, and we actually made it to the top of the mountain, which is about 2,500 meters. And it was easy, because from here, we actually took a ride, and we got up there. And sometimes we actually found accidental findings as well. I actually found that this is a Princess neutron monitor, which, according to my Wikipedia searches, has one of the greatest geomagnetic cutoff. I don't know what that means, but some of you might know. Anyways, and finally, I guess as I quote unquote mature or age, maybe I'll have the chance to look back and see if the research that we made is worth it to be revisited and to be a reference by others. And I was just talking to my kid after the, what do you call it, open house. She also went, and I was like, hey, are you sure you still want to do what I do? And because she always tells me that I want to be a scientist because it's cool. And she still says yes. And I think because being so conservative, I kind of go, are you sure? But then I look back, I'm like, maybe she's right, because we started this because we think it's cool, right? So I think that's what kept me going. And then along the way, I was watching some movie with my kids, and there was this Sing 2 cartoon, which is really great. U2, again, in my opinion, is a great song. Other people don't think so. It says, the song's title is Your Song Saved My Life. And I think I'm very grateful that I was at IMS, because along the way, sometimes I may be the lion or the monkey or the pig on the stage to give a talk, but the fact is that there are so many people behind me that make this work. And this, I definitely would say, the many mentors, the NIN, the staff that at IMS, with your support, that made it possible, and of course my lab member. I would like to thank all of them. Anthony Wood is the one who developed the Hongred recorder, and then Brian is the one who studied the big and the small vesicles, and Anthony's wrapping up the pumps for that right now. Jasper is the one who spearheaded the DNA repairs. And of course, we are very fortunate to have the many different mentors as collaborators. Due to time, I'm not going to go over, but I would like to thank everybody again for your attention and our funding to make this possible. Thank you.
Thank you for the interesting talk. And the floor is open for questions. Any questions? I thought you have found something you have been looking for, right? At least INS is the place, right? You want to spend the rest of your life here?
Scientifically, I'm still searching. Yeah.
Okay,
I later found that the song actually has a very strong gospel background. I didn't know that. But anyway, apparently, yeah, it's a relentless pursuit of science for me. Yeah, but also disappointed.
Okay.
Yeah.
Okay.
So this it seems to me this length scale of 200 nanometer to divide into small or big. It's rather arbitrary. It's some gurus of the EV, the first day when he did it, he used a filter of 200 nanometer, and then all the followers just follow that. But there's got, since you find some physiological evidence, there is indeed certain difference in their effects and behavior. So is there any particular understanding or reason why it has to be 200 nanometers? It seems to be too arbitrary to be a good number for dividing the size.
Great take is completely arbitrary. Yeah. So it's divided by us human, right? We think we're smarter, but no, you're totally right. It's completely arbitrary, and hence I bring this up again, that you see the overlap. Yeah, there is always overlap, and we try at least a year. We cannot separate them or get rid of them. It's always there. So I guess this, we are not the first one to show that the bigger ones has a function, but we are the first to systematically compare them and show that those two coexist. Why are you blinding yourself from seeing it, right? So I think from here on is that we're hoping to bring the awareness that people should look at more holistically, rather than just a small one. Even though the nano sounds more appealing, but that's not the whole world. So yes, you're completely right on the spot. It's arbitrary, and it's also very cell type specific. Not all cells release bigger ones. Yeah. So I hope that answers your question.
Well, my question is that is it worthwhile to pursue a sort of a fundamentally significant or from its functional point of view or whatever point of view that there is actually a meaningful separation of or this divide, you know, is there any, based on physiological or whatever function that can be much more meaningful than the filter somebody just used 10 years ago?
Right.
Is it worth pursuing to try to find that divide?
We're trying to show that. Yeah.
No, yeah.
So essentially, in terms of basic biology, I think it's critical that people realize that they have different properties. Okay. Then the next problem would be, what is more important, the small one or the big one? I think that's something that people need to look into as well. So yes, it is definitely worthwhile. But how far do we want to pursue this? We're trying that right now. The past year just a lot of hard work of animal experiments. We're doing the multiplexing to study cancer vesicles when they are co-treated, and we're seeing some interesting patterns. I think that's as much as I can say here. So yes, I think it's important. So I'm hoping that from here on forth, people don't only look at one population. In terms of application, it's actually a lot easier to produce the bigger vesicles because you don't need such a high ultrasonification or filtration effect, and people actually prefer that. Yeah, because I think the big one you only need 20,000 G, whereas the small one you need at least 100,000 G. Yeah, so people actually are thinking now, because when we started that workshop, people have reported therapeutic effect of the bigger vesicle, and the production of that is so much easier. Yeah, so there are definitely significant important implications on that. Yeah, but for my future research, I'm really interested about the Google Street View. I'm sorry if you're the vesicle people, there are so many vesicle products in Taiwan that's being advertised right now. If you want to buy it, come to me. Because, oh God, they don't have mechanism of action. They don't have quality control. They don't have release criteria. If I were you, I won't buy it. Just FYI. Yeah, but what I meant is that once you start to realize what cells they're affecting, where they go to, you can understand how they may work. And I think that's something that's largely missing in the vesicle field because they could not track it right. Yeah, and I think that's why I say we're asking more important biological questions after we have developed the tools. But of course, we're combining other technologies, such as the more recent I mentioned about cytometry by time of flight, and stuff like that to do, say, immunophenotypings to categorize all the different immune cells, such and such. We're also combining other imaging methods, such as Codex, or special biology, such as 10-time genomics and stuff. We're working into that, but we're preparing, yes.
You found out your EV are tumorigenic, right?
Yes.
Because you use the EV from cancer cells?
Yes.
So in principle, if you extract the EV from stem cell, they should be safe. Is that true? Or they may be tumorigenic as well?
Scientifically, we have not tested, so I cannot comment on it.
But the people must have done this experiment before, right?
Who?
People, other researchers.
People.
Because you just extract the EV from stem cells, right? And you just inject into the mouse, and you will see the result immediately, right?
Are you still recording?
Okay, let's
You want me to pause it?
turn off the microphone.
Well, I would say this, they have not been looking into dosage.
Why? This is a simple experiment to do, right?
I gave a talk at International Society for Cell and Gene Therapy in Paris last year. When I reported that the vesicle can kill,
Right.
the venture capitalists just go berserk. They say, I never heard about this.
Right.
And then the mesenchymal stem cell guy, one of the lady actually came up and said, this is not surprising because if you injected too many mesenchymal stem cell, it can also kill. And
But that's sales, right? But if
Right.
you inject EV,
so,
it's different.
so here's the thing. The fact that vesicles may kill was never on their radar. They were only driving a phenotypic effect, meaning that they were only driving a therapeutic observation, but they never care about the dosage at that time, much. I'm not saying that, so that's the reason why the US FDA, at least until April this year, specifically released a statement saying that there is no exosome approved product to date.
But the people in Taiwan are so excited about this product, right? I mean,
And hence I say, come to me.
okay.
And then I'll tell you, and then you decide.
Okay. More questions? Yeah, inside.
To be honest, I feel kind of strange. I mean, you, not just you, many of you working on biophysics are asking, where is the important biological question? But if you go to ask a hospital doctor, definitely they will ask a lot of important biological questions. For example, why Taiwan people easy to get lung cancer and so on. In my imagination, I would say there should be a lot of important biological questions. And you develop some technique and then you are asking this question. Maybe you should go to important biological problem question and then try to find some way to attack that question.
Yeah, so thank you, Vincent. This is really good point, and this is why I put that song on there. I struggle every day, but I am always exploring. I actually just finished a meeting with what do you call it, IDR, 工研院. Yeah, one of the 副所长, I forgot her, Maggie Lu. We were talking about the application and the potential translation. Yes, there are certain indications. 有一些病是很 well defined, and it's easier to tackle. 有一些东西是比较 complex, 像 cancer. 那 how I usually approach my research is to understand the fundamentals of it rather than the immediate application. And just coincidentally, we were recruited by the NSTC head, 那个我忘记他叫什么名字啊。然后我们被训了一顿。他说你们这些发明的研究东西根本就是 crap。我说 OK。然后他说你们以前这句话是我比较 shock 的，他敢讲说你们前辈根本都是 crap 嘛。This is public, so this is not secret. And 他就是like OK， 他说你们接下来要研究的东西应该都是可以显著的为台湾的人民做什么事情。然后不要玩花样。我就想到那个我们那个张昭炳讲堂外面有一个玻璃，上面说不要玩花样。我不是很聪明的人，但是因为我是一个很执着的人，因为我会一直问说这个问题重不重要。会玩花样的人大有人在，就是 OK， 我们就不要讲这一块。是为了发，因为他是这样讲，那个whatever， 他就是说是为了发 paper 也好，为了拿奖金也好，或什么。他说你们不要搞这种东西。我想说你要知道人家吃饭嘛，但事实上是在这个中间，我一直去取那个 balance， 就是说我今天还是往那个方向去。所以医生其实我认识几位，但是到头来其实 festival 遇到的一些问题我大概知道，不外乎是那个 scale up the production， 他的刚才有讲 release criteria 就是。FDA 为什么不给过？很简单，是像阿司匹林，你今天做这个药跟10年之后做的药要有同样的安全性跟药性。Vesicle 我很难连我自己出产的我都很难跟你讲这句话，因为它是会变的，因为细胞今天养跟10年之后养可能会有差别。那它要怎么样去做这个规定？所以 FDA 不敢乱给过来，不然会市场大乱。可是每个地方不一样，像日本的话，我前个礼拜才去。他们对这一块是还蛮开放式的，就是在某种程度上，医生他如果觉得 OK， 他就可以执行像那个 PRP 或者是干细胞的注射，他说 OK， 他不需要经过 FDA。这一点我有一点吓到。台湾的话就是我要就这个我不想在这边讲，就是啦所以是你的讲的没有错，我还是一直在 explore 这一块。那 我觉得我们可以帮助的是在 vesicle 的 mechanism 还有它的 quality control 这一块，或许可以协助他们更快的去发展出可能以后的产品。但很多东西大家都在摸，但是商人跟研究者的想法不一样。台湾尤其是亚洲的投资人的眼光比较及时。国外的深刻的投资眼光是以10年为单位，中间会有不同的 milestone。这个时候就，
台湾是年还月？
台湾大概是2年会问你说钱回来了没，有没有开始赚钱，3年就是说现在有没有啊，我抽了，那你就会发现说他们的那个 scale 不太一样。但是这个其实是他们国外他们已经知道那一套玩法。那日本其实说老实话也还在挣扎，要不然日本这么一个 advanced 的国家，他们 IP 的 C 到据我了解，他们有 Nobel Prize 的一个 laureate， 可是他们据我了解砸了很多资源下去。我上次才跟他们开会，他们其实遇到一些蛮棘手的问题。那今天这个场合就不讲，对啊。所以，但是他们现在也在往 vesicle 这一块想要转看看。但我就说，有时候其实回到应成您的问题，就是 biology 这种东西会很容易让人家误入歧途，因为中间会有很多 distraction， 就是叫你去开公司啊，或者
猫老师。
那赚钱是正确，只要是正当的。
那个也觉得我同意跟人说的，做 drug delivery 这个 EV 是不好的，因为很难做，它的生产什么都麻烦。但是我觉得有个领域应该一个技术可以有帮助，就是做 biomarker， 因为你可以完全 ex vivo 把它拿出来，然后用你的技术去帮忙去找。biomarker， 那也就是 vesicle 里面到底带了什么东西？那个东西它是一坨，它是一起发生的，它不是分开的，你比较容易去做一个大数据的整理，然后去 identify 是什么病，这样子可能是一个比较快的。
我有想过，谢谢莫老师的建议。我之前其实在马斯总医院的时候，有在做那个 circulating tumor cell 的 microfluidic strip 的合作。我们其实那时候有想要，就是像您说的，从血液里面萃取出脑癌病患。I'm sorry， 英文 ，Yeah，like we have collaborated on developing microfluidic strip to capture glioma cell release vesicle from blood for diagnosis. We succeeded in doing it. But the thing is that my method will help with preclinical study because we do require the donor cells to release to express our reporter. We cannot label already harvested vesicles. So this is a limitation of our method. And hence I said that it's more preclinical study per se. But what you mentioned is good, and in fact people have been working on it. That they have found that one of my former colleagues, Johan Scott, started a company that developed kits to diagnose prostate cancer from urine. And it was covered by the insurance companies in the States, which I think is quite a success. So people have been doing that. And probably not just the vesicles, they just isolate as much nucleic acid signature from the fluid for what you described. And it is definitely ongoing. Yeah, so it's a really valid point, and I think a lot of people are still using different methods to work on that. Yeah.
我想最后一点时间，我就帮品官回答应成的问题啊。从我的角度来看啊，那生物问题，我们生物学家一直说要找重要的生物问题，因为生物的跟物理化学的问题不一样的地方是 complexity 差太多了，是 level 差太多了。物理的系统你可以有统一场论 ，unified theory 解释了所有的现象，可生物学没有这个统一场论，统一的理论在这边。所以有很多种各式的diversity 太大，所以每个生物学家只能做一小块，一个很小的一个 domain， 那他其他 domain 他就不太懂了啊。我想这是我对生物学跟物理学的差异的一个了解啊，给大家做参考。More questions? Yes, the last one.
Hi, I have a small disclaimer. I'm chemistry, not biochem, so there's a bunch of stuff I don't really understand. First of all, I want to, maybe I didn't get this very well on how the EVs are injected into the rats. So what makes the biomarkers attach themselves onto the EVs and not other normal cells? Because they attach onto the lipid bilayer, is that correct? And well, another question that was raised by the other professor was, what exactly is, other than the size, is there really a difference inside on what the contents are inside? And also, what makes the EVs decide, oh, I'm gonna stay more in the lung than the spleen or the kidney? Like, what makes their decision on where to stay longer? And one last question would be, so since there's a difference on where the organs they stay in, would there be a difference on where you guys inject the EVs?
Okay, really good questions. Yeah. So let me start with the easiest one was what? The first one. Do you remember?
Why do they attach onto the EV's membrane and not onto a cell
I see.
membrane?
The reporter has a palmitoylation moiety. So this is borrowed from G-coupled receptor that is this is biology. It's an acylation moiety. Part of the acylation moiety. So there's palmitoylation, there's mirror stimulation. We took one part of it. It will just automatically insert into the inner leaflet of the lipid bilayer. Okay, so that's by nature's design. Okay, and then we borrow that and then we put it there. Okay, and then to your question about the biodistribution or whether there's specificity of the vesicles to certain organs, right? What we have found, at least in our case, if you express certain proteins, okay, it will target those organs, and this may most likely be receptor-ligand interaction. We didn't do detailed analysis on that, but we did do overexpression or knockdown experiments. What I mean by that is that we overly express the expression of the protein, or we lower the expression of those proteins. And we do find that it significantly affects their distribution in the body. So chances are it's receptor-protein interaction on the surface of the recipient's tissue, as well as on the surface of the vesicles. Yeah, but we didn't do extremely detailed like biophysical examination of that interaction, which is a fun part to look into from a biophysical perspective. Yeah.
So when the cells build the vesicles, they implicitly have an idea of where they want to send this vesicle to based on these.
Having an idea is a very subjective thing. I think it's evolution. Whatever worked the best for the disease, they will do it, and that's cancer. Cancer is essentially evolution. On the other hand, you do pique my interest, which we have been trying, at least I don't think my student know this. I have been very interested about the homeostasis maintained by the vesicle communication. It's like today's world. We're all friendly, we talk to each other. If there's any misunderstanding, we talk it out. This is what I would call in the term homeostasis in animal. I think vesicle does play a role doing that. But studying that is very difficult because there is no disease phenotype when you study it very well. But it is something that we're trying to look into. Yeah, so your question is how do they know to express what, to go somewhere? I think it's just randomness, selection.
Yeah, I think random process.
Yeah, but it's a very interesting question that I think it can be studied. I don't know if I can live that long to study it, but I think it's cool, yeah.
One last quick question. Sorry, my bad. Because I always thought that cancer cells would move like the entire cells themselves through the bloodstream. But now you said that they use vesicles to transport their information. So are they like infecting our cells and making them cancerous? Is that what you mean by that?
Yeah, it's like in a war right, between Ukraine and Russia. Before the war actually started, like they sent a ton of spies. Yeah, go back to the media and see what happened before the actual war happened. There were a lot of things going on inside Ukraine. So vesicles were sent by these cancer cells before the actual invasion to the secondary or tertiary site. This is one theory that was shown in animal study, but because you shouldn't do human experiments, right? So at least in animal, that's what they show. They show that cancer cells release those vesicles to tertiary or secondary sites before the actual migration, right? So that is one theory. It could actually happen at the same time. We don't know yet. So such that they send the soldiers and they send false information at the same time. So that is also possible. And I think for cancer, don't limit your imagination. I think they will do everything to survive. Yeah. So yeah, keep it open.
Thank you.
Okay, I think it's time to stop here. Let's thank the speaker again.
Thank you.